We start the session with Matthias Hentze, who is Professor and Director, European Molecular Biology Laboratory, EMBL. Matthias did his MD at University of Münster, and his postdoctoral training was at NIH. And in general, he and his group have been very much interested in looking at RNA binding proteins of different kinds, the riboregulation of these proteins in health and disease. So with that short introduction, it's all yours.
Good afternoon. Welcome back after lunch. After the two beautiful presentations that we had this morning, which were very, very stimulating. Now it's my job to keep you from, postprandial, fatigue. And the way I will try to do this is actually, cover a topic that, unlike condensates, where I'm pretty sure everyone here in the room had heard about condensates before, I'll talk about riboregulation, which I know a few of you already have heard of and maybe familiar with, but perhaps others are not. So the way I have divided my two talks is the first one is called Riboregulation, the Story Begins. So that's sort of an introductory part. How do we see it? What does it do? What are some of the examples that have been characterized? What have we learned from them? And then in the second part, we'll get there. Okay, so, riboregulation is a term that we did not start or that we did not introduce. In fact, the term riboregulation has been applied in the past in sort of various settings. Usually, what these usages have in common is that RNA is a regulator. And so the most common past use of it has been, for example, in the context of microRNA regulation, where quite obviously, small microRNAs are the regulators of messenger RNA expression. So my usage of that term does not relate to this. In fact, I don't think it relates to exploiting the base pairing potential of RNA for regulation, and in particular for regulatory specificity. I mean, where we have base pairing, we have a beautiful principle of how specificity is achieved. In what I will be covering, the question of specificity will be an interesting one to discuss. So let's start, nice and easy. It was already said in the introduction that, one of our key interests is in the field of RNA binding proteins. And I would say, this slide from a personal perspective also sort of covers my first 20 years of being a PI, when our lab was working on RNA binding proteins that bind to RNA and in particular played roles in messenger RNA translation and messenger RNA stability in the cytoplasm. More generally speaking, what this slide introduces is sort of the two main areas where many of the RNA binding proteins we were familiar with are active. And that is along the entire gene expression pathway from transcription to RNA decay and everything in between as sort of one main area of where RNA binding proteins are important. And the second area is, of course, as components of RNP machines, such as the ribosome, the spliceosome, telomerase, there are others. So these are sort of the classical, I would say, fields in which RNA binding proteins have been studied. But when I speak this afternoon about riboregulation, I'll actually do that in a different context. And so again, thinking about very general principles, very much about in biology that we want to understand and the regulation in biology that we want to understand can be reduced to regulation of protein-protein interactions, or in the case of metabolism, to the regulation of enzymes and potentially enzyme-substrate interactions. And you're well aware from the vast published literature that very much of this regulation can be exerted by different forms of post-translational modifications being applied to proteins and regulating aspects of protein-protein interactions, as well as regulating enzymes. And I'm just showing 3 forms by way of example of PTMs on this slide. So when I talk about riboregulation, what I mean is the idea, the principle, that RNA can do a very similar job. That RNA can, by means of directly interacting with proteins in a biologically meaningful way, alter protein functions and molecularly do so by affecting aspects of protein-protein interactions, protein fate in other ways, or in the case of enzymes, enzyme-substrate interactions. And I'll discuss examples of that. So, how did all of this come about? It really started with experiments that my lab was doing, and in parallel, the lab of Markus Landthaler at the MDC in Berlin were doing in parallel. And that came out of, I would say, the end of the first decade of this century. If you think about what was the highlights of that phase of science, one of the things one would definitely name is of the area of omics, of systems biology. Starting with the sequencing of the genome, transcriptomics following, proteomics following, metabolomics following. It was omics sort of all over, collecting parts lists. And as an RNA biologist, and an RNA biologist being interested in RNA binding proteins, of course, we were very much driven by the question of what might all the active RNA binding proteins in an organism or in a cell be? And could we devise experimental approaches to do so? Of course, one could go about it computationally and look for RNA binding domains, in previously unknown open reading frames, and predict their possible function as RBPs. But we wanted to find an experimental approach. And the beauty, at least in my eyes, of molecular biology is that the individual tools, I'm not talking about high technology here, I'm talking about sort of the ground toolbox of molecular biology, has oftentimes very simple tools, and you can combine them in novel ways, and that way find novel solutions and devise relatively simple, new yet very powerful technologies. And I think this turned out to be an example of this. So in order to try and define the RNA binding proteins, initially of HeLa cells, we used UV cross-linking with 254 nanometer UV light. That had been established in the 1970s to introduce covalent bonds between proteins and single-stranded nucleic acids, and only so when there was an immediate contact between the two, because the reaction chemistry is such that it is very short range. And UV cross-linking does typically not lead to protein-protein cross-linking. And so in this way, we could introduce, if you take these 3 eggs here as RNA binding proteins, and this as a polyadenylated messenger RNA, we could introduce a covalent bond between the red egg, which is the immediately touching protein, but not the yellowish eggs, and establish this covalent bond. Well, then cells could be lysed, and again, using a very old technique, oligo-DT selection of polyadenylated RNA, we could pull on the poly A tails, with it pull the cross-linked proteins, and then use very stringent washing to remove anything that isn't covalently bound. You can take an oligo-DT RNA hybrid and really wash it harshly with high salt washes, with detergents, with denaturing agents, et cetera, et cetera. So you get a pretty clean result at the end of that washing, and then you release the cross-linked protein that has also been denatured. So the egg has turned into a sausage, and that is released with RNase treatment, and then the sausage protein is identified by mass spec. And that very quickly, within the first set and round of follow-up experiments, expanded the candidate, I should say, RNA-bound proteome from, for a mammalian cell, 7 to 800 to easily over 2 and probably over 3,000. And so that was very surprising. And, it was actually not only pleasing, but it was also worrisome. And I'll come to that in a couple of slides. I just want to interject that, we call this approach that I just illustrated to you, and it's also shown here on the top, RNA interactome capture. As I said, the lab of Markus Landthaler did very similar work with very similar results, exactly in parallel. But then subsequently, our lab and others have developed other approaches, I would say pretty much equally simple, with detailed advantages and disadvantages that I won't take the time to discuss, at least now. In this case, it is using, the silica columns that every RNA lab, nearly every RNA lab has to purify RNA from cells. Claudio Sensio found in our lab that when you UV crossing before, that works by a similar principle. It is not limited to polyadenylated RNAs, as the oligo-DT capture is. So that's clearly an advantage. The stringency of this approach, as well as this one, where you use organic solvent extraction and sort of use recover the interface where RNA-protein interactions are. The stringency, I would say, is slightly less in this case, but you're not limited by the nature of the RNA. And then there is an approach where you introduce modified nucleotides like ethyluridine into the RNA, into the nascent newly made RNAs, and then using a click chemistry, put on a biotin and use streptavidin beads to again pull out cross-linked RNA binding proteins. So the common denominator of all of these approaches is the use of UV cross-linking to covalently capture the direct RNA binding proteins on the RNA. Okay, so maybe a final view sort of the picture that I'm presenting. What you see here is sort of the same type of RNA binding proteins that were on my first overview slide. Classical RNA binding proteins that often have one or several RNA binding domains, globular RNA binding domains, through which they bind RNA. And then as a consequence of that interaction, usually not just a single protein, the fate of the RNA, its stability, processing, translation, is being altered. In other words, the protein exerts, the RNA binding protein exerts a dominant regulatory function over the fate of the RNA. In riboregulation, this picture is exactly turned 180 degrees around. Here, the RNA binds to the protein and is thought to change the protein's fate. As you will also see as we go on, the list of these RNA binding proteins is very different, from H&RP proteins or KH domain RMs type of RNA binding proteins. Here, we have typically proteins that do not have any recognizable RNA binding domain. hence also the question of specificity. Yet, as I will show, in very specific interactions, RNAs bind to proteins and alter aspects of protein function. Now, that idea, perhaps as presented sort of broadly as a broad concept, may have been new to the work that we and others were doing subsequent to the work I just summarized. But, you know, clearly there were examples in the literature that when you look at them, you know, you actually realize these proteins follow that principle. So, for example, we were talking this morning about the integrated stress response. You know, one of the proteins that was on Christine's slide is the kinase PKR, which is one of them, which is stimulated by double-stranded RNA and then phosphorylates eIF2-alpha, as you know. So this is a classical example of what I'm saying. RNA alters the function of this kinase from an inactive to an active state, you know, in a biologically meaningful setting. Similarly, in E. coli, there is a small non-coding RNA that controls RNA polymerase work exactly around the turn of the century, or the toll-like receptors. I've copied here a slide on toll-like receptor 3. As you know, there are other toll-like receptors that are being activated by RNA, as are a number of other examples in the innate immune response. So, we were set with this long list of proteins where you had to worry, is it really that the RNA-protein interaction that we are scoring by the assays that I illustrated to you, that made them candidate RNA binding proteins? You know, is this really something that hides meaningful biology? Or is it perhaps something that is driven by certain, you know, random interactions of abundant proteins that are then trapped by the UV cross-linking, you know, charge patches that, you know, help with this? You know, clearly these long lists of RNA binding proteins did not mean to us at the time, nor today, that, you know, these are all important, biologically important RNA-protein interactions. And unfortunately, I still believe today we need to explore the functionality or the functional relevance of this still pretty much in a one-to-one way, although we are currently having some projects in the lab to try to change this, but I will not cover that today. So anyway, you know, just following this, a postdoc, Rusty Horos, joined our lab, you know, and discussing a project. I said to him, why don't you take this very, very long list and, you know, you just choose any protein you wish, you know, the one that intrigues you the most. The only criterion it needs to fulfill is that it should be a protein where already a lot of work has been done, as evidenced by having many entries, we said over 1,000, in PubMed. And when then one does text mining on what's deposited in the literature, there should not be any evidence that that protein was previously implicated as an RNA binding protein or implicated in RNA biology. And the protein that Rusty chose is, is the protein P62, or an alternative name is sequestration 1. I think I also found sequestration 1 on one of the slides this morning. You know, P62, as I will call it, had very many publications already to its credit. Many of them were for its role in selective autophagy. And the function of P62 in autophagy is that in response to an autophagic stimulus, such as starvation, P62 monomers homo-oligomerize and build a bridge between the forming autophagosomal membrane at the one end and the sort of cargo that is to be degraded, eaten up by autophagy at the other. So a very important adapter function in this sense, driven by this homo-oligomer. And what Rusty then found was he took this protein that does not have any recognizable classical RNA binding domain, and he had identified by clip approaches, crossing immunoprecipitation approaches, that the RNA that binds to it very tightly is a small non-coding RNA transcribed by RNA polymerase 3, so it's tRNA-like. And the function of this RNA-protein interaction is the RNA blocks the protein from forming oligomers. And so in response to a trigger for autophagy, starvation, the levels of this RNA are downregulated. So monomers are freed, they can oligomerize, and sort of autophagy can proceed. That obviously raised questions. What about, what makes this interaction, a specific one? I wouldn't say the answer will be out before we have, proper structural, high-resolution structural information. But what we know from mutagenesis and functional studies is that at this very N-terminal domain of P62, which, as you can see here, in homo-oligomer formation, involves the interaction between subunits of positively charged region that involves this arginine 21 and a negatively charged patch a little further C-terminal, that Volt RNA 1-1 interacts with around arginine 21, a single amino acid exchange, arginine to alanine, will prevent this RNA binding and will prevent the inhibitory interaction. And if you take the Volt RNA 1-1, there are 4 Volt RNAs in the human genome. There's lots of tRNAs, of course, in addition. And this particular one has this rather large central flexible loop, and single nucleotide exchanges within this loop will also destroy that interaction. So it appears that these are critical determinants of specificity here. Okay, the second example I want to give you is arrived from this kind of picture. What you see here is the glycolytic pathway, from glucose to pyruvate. And you can also see that, when an enzyme is annotated with a green dot, it means that that particular enzyme was found to be RNA binding. So a short summary, not exactly 100% accurate, but a short summary would be every enzyme of glycolysis is also an RNA binding protein. Now, could that be? I mean, we have been studying, not we, but scientists have been studying glycolysis for about 100 years. And certainly, roles of RNA of that type had not been observed. So that was, again, very intriguing, a challenge that Ina Huppertz took on. Specifically, she studied the penultimate enzyme of glycolysis, which is called enolase 1. And so what she did was, again, she used clipping, cross-linking, immunoprecipitation, and then sequencing of the library to determine, what are the RNAs that bind to enolase 1. And unlike the picture that we obtained with P62 and Volt RNA 1-1, here the picture was totally different. There was, a small ocean around 2,000 RNAs that specifically enriched. They were mostly regions from within messenger RNAs, mature messenger RNAs, usually 5' or 3' UTR regions. And you can just see 6 of them annotated here because those were the 6 that we sort of took from the representative center of this cloud for functional analyses. So the first thing that you want to know is the binding, specific, and what's the affinity of the interaction. So this is a band shift assay using recombinant human enolase 1, and one of these RNA regions from the cloud, which is from the 5' UTR of the poly-A binding protein C1 encoding messenger RNA. It's a 35 nucleotide synthetic piece of RNA, so it's 35 nucleotides, and you can see here, titrating in, increasing amount of RNAs. You can see a nice band shift. You can see that when we use a control RNA, the control RNA is a piece of 35 nucleotides that has a GC matched content, but does not show a peak in the clipping. And that's what we use as a negative control. And you can see there is no such, sorry, I'm misspeaking here. So sorry, I'm confusing you probably by this. I take it back. This is a competition assay. So we're using the wild type probe, and then we use unlabeled RNA as a competitor that is being titrated in. And so as you can see, and as I said, you see a nice shifted complex that then disappears as we titrate in the cold competitor. But when we titrate in the control RNA that has the characteristics that I mentioned, GC match control, you can see there is no such competition. And you can see that in terms of affinity, there is at least a factor of 100, if not more, difference between the wild type and the mutant. So this is highly specific. And when we used other probes and other competitors, very similar parameters were obtained. So there is clear sort of in vitro specificity to this. But the question is it functional? So as the next thing, we took the recombinant protein, again, E. coli, purified human enolase 1, and added the very same 35 nucleotide, we call them ligand RNA regions, using 3 different ones. Again, using as controls GC matched equal length controls from the respective RNAs. And as you can nicely see, here's the enzymatic assay curve without any RNA added. And on top, the 3 control RNAs added. They make absolutely no difference whatsoever. But when you use either of the 3 ligand or target RNAs, you can see that the enzymatic activity is inhibited, giving a very nice reflection of riboregulation in vitro in a pure biochemical assay. So what about cells? In order to address this in cells, Ina took mouse embryonic stem cells. The reason of that choice will become apparent in a second. And she now nucleofected, again, either the control RNAs or the specific ligand RNAs. And as you can nicely see here, in all 3 of the 3 cases tested, she observed a significant reduction in lactate production, which was our readout for glycolysis. So showing that apparently there's also a specific inhibition of glycolysis by RNA via enolase 1. I say that because we've also tested a mutant that doesn't bind RNA anymore, through these ligand RNAs. Now, that shows that RNA can do this when heavily over-transfected into cells. But is that really used in biology? So in order to address this, and this is also some common technical approach that we have been using, we like to make mutants of the wild type protein, where the mutant should still be able to fulfill other known functions, in this particular case, be active as an enolase enzyme. But the one thing that's being changed is its RNA binding. And Ina was actually lucky to obtain a mutant that showed increased RNA binding compared to the wild type, about twofold, as well as decreased RNA binding, about down by a factor of 3. And as I said, very importantly, when you take these mutants and compare them with the wild type for enzymatic activity without any RNA added in an in vitro system, they show very comparable enzymatic activity, showing that the mutation hasn't changed the enzyme per se, but changed its RNA binding. And when Ina now engineer these single point mutations, this is a single amino acid exchange. You will see it on the next slide. This is an exchange of 3 amino acids. So when now these mutants were engineered into the genome of mouse embryonic stem cells can be triggered in vitro to differentiate. It's a very simple protocol to do that. And when you do that, the metabolism of these stem cells changes, as is quite common in stem cell biology with stem cells undergoing differentiation. So here, pluripotent stem cells, which are very active in glycolysis, down-regulate their glycolysis, and respiration comes up. And when we now prohibit the proper functioning of the riboregulation of enolase from these mouse embryonic stem cells, you can see that both in the mutant that shows constitutively increased RNA binding, as well as in the mutant that has a decrease in RNA binding, there is disturbance of stem cell differentiation. So showing that in this case, riboregulation RNA serves to control an enzyme function in a biologically meaningful way. And this is not any structure that we have of this. I guess you recognize this immediately from this. But this is just the PDB structure of enolase as an enzyme in its active state, where in mouse embryonic stem cells, the RNA binding activity of enolase is low. When stem cells are then stimulated to undergo differentiation, the RNA binding activity is activated. This activation happens through the acetylation of these 3 lysines here, K105, 89, and 92. We know that from mutagenesis. And then we surmise may open up, access to the catalytic center of enolase for RNA to bind. Our strongest evidence that this is a region important for RNA binding is that the RNA down mutation is this lysine 343, which is right at the catalytic center. When we change that to alanine, RNA binding drops by at least 1 order of magnitude, and as I've shown to you, results in a loss of riboregulation. So in this particular case, we have 2 differences from the first example that I shared with you. The first one is that in the first case, it was regulating the amount of the RNA regulator, the abundance of the RNA regulator, volt RNA 1-1, in a physiologically meaningful way, which would then result in riboregulation. In this case, where we have 2,000, as I indicated, RNA ligands across the transcriptome, we think that this is a highly redundant regulatory system. It's the collective of the binding sites that are available at very high concentration, as you can imagine, which result in the regulation. We call that, like crowdfunding is sort of a collective, funding approach. We call this crowd controlling. And then when through activation of the RNA binding activity, inhibition occurs. As I said, it's not by a single RNA, but it is by a collective. So, this basically just summarizes everything I've mentioned to you by way of introduction. And, as we move forward with this and other things, I believe that the systematic exploration of riboregulatory interactions, I showed you some mutants. These mutants, both in the case of P62 as well as of enolase, identify, critical regulatory interaction sites. And I would imagine that this type of information can serve in the future to develop therapeutic approaches in cases where those proteins play important roles in biology. P62, for example, in some of the neurodegenerative disorders. Enolase is a cancer target, a validated one. And many other proteins on the list have their own. So I finished this part. This is just, if you want to read up, not only about these examples, but there are by now very many others, really exciting examples of riboregulation out there. There is one example where we know the structure of the regulatory RNA-protein complex. In the case of SHMT1 messenger RNA, that is in amino acid metabolism. It's a beautiful work by Francesca Cotrosola from Rome that was published last year. This and others are also covered in this review. And so this is my last slide of this. Just to, we started easy, let's finish easy. While I think that sort of in the classical way of looking at RNA binding proteins, where the protein sort of exerts, where the protein exerts a dominance on the RNA and regulates the RNA, riboregulation sort of turns that picture around. So I think we can view RNA-protein interactions sort of not as a game of dominance, but as a beautiful pas de deux. So I finish with this. Happy to take questions.
All right, thank you, Matthias. We have time for questions. Do you have a mic around you that you can grab?
Hi. For the 3 RNA ligands that you talked about, among the entire RNA ligand pool, is there any sequence similarities that you see?
That's a very good question. So what distinguishes the pool of ligands from the pool of GC matched controls? Is there a motif you can find? So we have our own bioinformaticians as well as some others we've contacted have looked through it. We haven't found anything that we found a very convincing explanation of this. We've been looking at sort of secondary structure prediction programs that hasn't yielded anything that has really convinced us.
Which actually brings me to my second question.
Just one last sentence. So we think in order to understand it, we'll need a structure. And then, structure between one or more than of the ligands should hopefully decipher the denominators of specificity.
So for the concept of riboregulation, do you think that the secondary structure of these RNA ligands play a more role than the sequence, or is it the structure or the sequence which kind of governs regulation?
So obviously, I've stated what our experimental information is at this point, so I can only tell you what my interpretation is at the moment. And so that brings me back, actually, your question to one of the opening statements that I made about microRNA-mediated regulation, CRISPR, the CRISPR mechanism also, where base pairing is the key to the specificity. Here, I think, it may actually even come up at a later slide, doesn't matter. Here, I think we need to remind ourselves of the aptamer field. So I think what aptamers demonstrate is that you can evolve, you can select an RNA that binds with high affinity and specificity to a biological or even non-biological surface because of the RNA's ability to be very plastic in terms of its structure and evolve specificity that way against something, including a protein, that didn't itself evolve to bind RNA. So, as you can hear from my answer, it goes in the direction of the structural aspects of it.
If these RNAs are non-specific, do these RNAs code for any protein and then they're sequestered by these metabolic enzymes and thereby reduce the translation of those RNAs, and it can be a secondary effect and not a direct effect of what you're seeing? So how do you negate that? But yeah, it's
so the first part is to gosh, I'm still slightly jet lagged, I have to admit. Never mind. So you asked about whether this is a direct or an indirect effect, and maybe it's the enolase binding to these RNAs, changing their translational stability or whatever. That's a very relevant question. First of all, I don't want to imply that when the protein binds to the RNA that nothing would happen to the RNA, that riboregulation and what I would call moonlighting are mutually exclusive ways of an RNA, or ways how an RNA-protein interaction can affect biology. I think the two can go hand in hand and come together. But in this particular case, we knocked down enolase 1 and looked at mRNA by transcriptomic analysis at changes in mRNA levels or splicing, and by ribosome profiling, whether we could score any translational changes, and there was nothing that we could see. So the suggestion is it doesn't work in that direction. Yeah. Have I answered? Yeah.
Yes, thank you.
You told us that nearly every glycolytic enzyme is able to bind RNA. Then you described for enolase that there were thousands of mRNA sites that is recognized. Do the other glycolytic enzymes also respond to the same 1,000 mRNAs? If not, how would you explain the change in the whole pathway during differentiation? I mean, how does is this all coordinated?
Yeah. So there are, again, different parts to it. The first one is we did metabolomics also on the wild type scenario and on the mutant down scenario, where specifically the enolase block, as it were, by RNA is removed. And what those metabolomics show is that when the RNA is binding to enolase, there is sort of pretty much what you would predict. There is a reduction in pyruvate formation, which is downstream in the pathway, and there is an accumulation just above, and siphoning off into the serine pathway. So the consequence of this is not just to reduce pyruvate formation, but actually drives from the metabolomic data side pathways in this case. Now, your question was about all of the different enzymes. As I said earlier, I think we really need to study them one by one. And we have currently a project that looks quite deeply into PKM2, which is the last enzyme in the pathway, just following enolase 1. What we are seeing there is that from the RNAs that we identify by clipping, there is an overlap. and we can validate this overlap in terms of binding. We don't think that the mechanism of RNA-protein interaction is the same as here, like an obliteration of the catalytic center. We see more PKM2 forms in its active form tetramers. We think the RNA may affect tetramer formation, or at least the steady state pool size of tetramers. But we also see some RNAs that are not enolase targets. How that plays out, maybe also with additional enzymes in terms of network regulation, etc. That's exactly the key question that, before I retire, I hope we can get some more insight into.
I'm remembering Vault RNA. I thought Vault RNA was incredibly abundant and bound to proteins in some complex, which is incredibly abundant. Is that true? And how does this role for your RNA fit with that?
Yes.
RNP complex.
So what you're remembering is also what I remember. There is a complex called the Vault complex, which is giant, 3-4 times the size of the ribosome, made up of the major Vault protein and a couple of other associated proteins. In humans, it binds this Vault RNA 1-1, as well as the 3 other Vault RNAs. Hence the name Vault RNA, because it is associated with this particle. So we have been looking into the question, is it the naked RNA that is doing what I described, or is it maybe a particle function? We've knocked down the major Vault protein and that way destroyed the Vault complex, and that has no negative effect whatsoever on this type of riboregulation that I showed. And the other 3 Vault RNAs also don't seem to be able to substitute. So in this case, as I said, that's why I chose also the 2 examples. In one case, you have many, many different RNAs that can redundantly do the job. In this case, there's only one that we know.
That was really interesting. Your P62 result is particularly surprising to me. I hadn't seen that data before. So P62 is a marker that is often, you often see P62 inclusions in patient tissues of various neurodegenerative diseases, Alzheimer's, ALS, FTD, Parkinson's, Huntington's. And so it's always been presumed that that P62 was there because it's binding, quote unquote, misfolded protein. But you raised the interesting idea that maybe it's sequestering RNAs. So I'm just wondering whether or not you're thinking about digging into that. And is there an approach that one could experimentally try to figure out whether or not there are RNAs associated with those P62 inclusions?
Mm-hmm. Yeah, so before your slide this morning, I was aware of this sort of implication of P62 in this part of pathology. We haven't looked into it. As I think I said in passing, we think that the high-ish affinity binding that we see for the Vault RNA 1-1 is really quite a limited and specific one. So it doesn't have kind of this broad RNA binding as many proteins have that do that through intrinsically disordered regions or so. I'm not dismissing the idea. I'm only saying we haven't followed it, and I have sort of one consideration for why maybe it is possible, but not something I consider necessarily very likely for whatever that's worth.
So you showed us the data for eukaryotic enolase. Since I was a student, people have observed bacterial enolase as part of the bacterial RNA degradosome, and people didn't quite understand why enolase purified with an RNA degradation complex. Do you think now there is more clarity on that? Is it on the same lines of bacterial enolase binding specific mRNAs and aiding, I don't know, degradation or things like that?
So I've thought, I'm aware of this, and I've thought about what one could teach us about the other, but I haven't really come up with much clever of either why what we have observed might say instruct aspects of the E. coli degradosome, nor work on the degradosome, instructing things regarding what we see with our enolase. Because as I said, we've really been looking for evidence of changing stability of the RNAs that enolase interact with, and really haven't been able to find anything whatsoever.
All right, last question, and then we'll move.
Very nice talk. So you are talking about this, the enzymatic activities controlled by RNA. Have you seen that these RNAs are modified when it is interacting with such enzyme? My question is that whether RNA modification could control such activity.
Yes. So since I've been referring to this list of over 1,000, unexplored, well-known proteins with unexplored RNA-protein interactions, I would be pretty sure that this list will include aspects where the RNA-protein interaction is influenced by modifications of the RNA, such as methylations or other forms of modifications of the RNA. In the two examples that I discussed, we have been seeing, at least where we've been doing in vitro work, the effects on chemically made, unmodified RNAs. So those clearly don't seem to be necessary, for these in vitro settings. Whether in life and maybe in aspects we haven't studied, we only studied in the case of enolase, mouse embryonic stem cell differentiation. We haven't looked into other forms of stem cell differentiation, for example. There could be other mechanisms involved. I would definitely see space for that in the examples I discussed, or more likely in others.
Okay, with you, we can continue this thing.
Let's get on. We just need to. Okay, so now the story isn't beginning anymore, it now unfolds, as you can see. So what I want to do now is relatively quickly cover another example of riboregulation. So, proceeding on the subject that we've just been discussing. And then in the second part, talk about something which definitely is far from textbook knowledge, let's put it this way. It's quite early work, I say that right away. But I share with you what we know and what we don't know. And also why I'm very excited by it, because if that continues, I believe it can really, become a new, bubble in the RNA biology field and beyond. But you'll see why I say that. Okay, so this is where we left off, and this is where we are going now. This is another very important enzyme, in this case, a complex. It's the ATP synthase complex, well familiar to all of you, because that's where all our mitochondrial ATP comes from. So it's a critical enzyme. I read, but I haven't researched whether it's true recently, that the daily production of ATP by ATP synthase would be 70 kilograms per day. That number seems unbelievably high, so I must look into it. In any case, this is, as you know, the very important protein complex that's critical to our lives. And our list had implicated, these yellow mitochondrial matrix subunits as RNA binders. So this is my link to India, at least for today. We have a few really superb Indian colleagues in the lab. But this is Andrila Chatterjee. Andrila joined the lab, having worked on an aspect of mitochondrial metabolism for her PhD as a postdoc. And so she proposed for an EMBO fellowship to work on why, what is the biological relevance of these three subunits binding RNA. And EMBO fellowships, you have an interview with an EMBO member. So she had that interview, and the EMBO member said to her, look, I'm really impressed by the work you did for your PhD. You seem to be a very smart and productive scientist, but the project you're proposing is crap. I mean, that's an artifact. You know, the Nobel Prize for ATP synthase was given in 1997, you know, providing detailed structural information for how it functions. There's just no space, you know, for RNA. So, you know, I'm sorry I can't support you, you know, for the fellowship. She didn't get it. Fortunately, I found an alternative source for her, and she could, you know, proceed with a project that I'll quickly summarize. And maybe I'll start doing so by just referring to this other technique that we call complex capture. It's the silica column-based technique, you know, where, as I said before, you UV cross-link to covalently bind RNA to an RNA binding protein, if it is an RNA binding protein. It's then retained on the silica column. You can then elute it, and then you can use the eluate to either do mass spec or to look for particular proteins of interest. So in our case, you know, these 3 ATP synthase subunits. And so this is an immunoblot, as I already said. The first 2 lanes are input lanes, and then these middle lanes and the right-hand side lanes are eluate lanes. In the middle case, it was plus UV light. In the right side lanes, it's minus UV, so it's a negative control. You know, if the protein binds, because it's an RNA binding protein, it should be retained through RNA. And so you can see this is completely empty for all of the proteins. You know, then here you have histone H3, which is a DNA but not an RNA binding protein. And you can see even after cross-linking, you know, it isn't identified, even though it's a very abundant protein. Here you have an H and RNP protein as a positive control. And you can see it, and the other 3 subunits of ATP synthase show a similar pattern. So this supported our earlier data that it is indeed RNA binding. So as we go on, I'll just speak about this 5A1 subunit. And Andrila next proceeded to determine the RNAs that it binds to. And there I had my first surprise. You know, my expectation was that we would probably find mitochondrial RNAs as the ligands or as the interacting RNAs. That is hardly the case. And what really stands out is sort of around 250 or so, 220 cytosolic messenger RNAs as the ligands. Now, a mitochondrial matrix protein and cytosolic RNAs, does that make sense at all? Well, as you know, most of the mitochondrial proteins, including this, are nuclear encoded, are translated in the cytoplasm in precursor form, and are then imported into the mitochondria, proteolytically matured, and then they find their final, you know, destination and where they do their jobs. So the interaction of this protein with cytosolic RNA suggested that the interaction might happen before the protein, the pre-protein, enters into the mitochondria. So that made it, well, this shows, you know, some validations by RNA immune precipitation in qPCR. These were some of the ligands we tested. These were RNAs of at least equal abundance to these. Here we made recombinant protein and did a band shift assay. In this case, it's actually, like I had said falsely with the enolase, here it's a target RNA as a probe or an RNA scramble. And you can see, you know, nice specific band shifts with an apparent KD that is sort of in a range that a biologically meaningful interaction would be quite plausible. So then, obviously, the question was, where does the interaction happen? And so in order to address this, Andrila adapted a proximity ligation assay. In proximity ligation, as you probably know, when two arms are close enough to one another, a rolling circle amplification is enabled, which then creates the binding site for a fluorescent dye, and that way you can, show the proximity of the two arms that you've tested. So in this particular case, she used an antibody against the ATP 5A1, shown here, and she used an antisense oligonucleotide, not as shown here, I'm sorry, against the poly-A tail, but against the specific RNA that we had determined by clip, and that was biotinylated at the end, so providing for the second arm. And so then here you see super resolution microscopy using TOM70, which is the outer membrane protein 70, as sort of the gray outline of the mitochondria, showing in cyan the mature protein where it accumulates inside of the mitochondria, and then showing in red the proximity ligation assay for the interaction. And what you notice, and I think it's hopefully dark enough even with this slide, you notice the signal is exactly, the centroid of that signal is exactly on the outside of the mitochondrial membrane. So that was very much in keeping with cytosolic RNAs being the ligands. And it suggested to us that maybe this RNA-protein interactions could have something to do with the import of the precursor into the mitochondria. So we conducted a in vitro mitochondrial import assay. This is a very old school assay where you purify, actually, and we don't have the ability to do that, but Heike Rampelt, a young PI from the University of Freiburg, is an expert in this. So you purify human mitochondria. Then, because we wanted to learn about the role of associated RNA and its potential importance, those purified mitochondria were either RNase A treated to destroy associated RNA, or were mock incubated. The mitochondria were repurified, and then the import assay is done by in vitro translating the precursor protein, incubating it, and then following its mitochondrial import by following the proteolytic maturation of the protein. So that's how the assay works. This is the outcome. You see 3 minutes of import and 10 minutes of import. This is mock treatment. This is RNase A treatment. And you can see the import is less at 3 minutes after RNase treatment. The import difference is even larger after 10 minutes, suggesting that the RNase A treatment has removed RNA that was helpful to import. Or alternatively, it has somehow messed up the mitochondrial import machinery. And so to address that artifactual outcome, as it were, we took another enzyme that moves to the mitochondrial matrix by a similar import pathway. And you can see that here RNase treatment has absolutely made no difference to mitochondrial import, suggesting that, as I said, the RNA may facilitate the mitochondrial import of this precursor. We then made mutants, and we tested those mutants in RNA immunoprecipitation qPCR assays. And you can see that these 3 mutants, here you can see them detailed, all 3 show diminished RNA binding. We then tested those mutants because, as I said, when you make a mutant and you want to affect RNA binding, you want to make sure you don't mess up the protein overall. So the way this was done here was by knocking down the endogenous protein and then measuring oxygen consumption rate, the so-called seahorse assay that measures the complex function. And you can see here that after knockdown, the OCR is reduced. And then we did rescue with a wild type protein that's obviously insensitive to the RNAi. And you can see that it nicely rescues the situation. And you can see that all 3 mutants equally well rescue mitochondrial respiration. So that tells you 2 things. First of all, the RNA binding that we saw is definitely not absolutely required for getting it into the mitochondria. You can see here all 3 mutants that show diminished RNA binding, beautifully rescue the OCR. But it shows also that these proteins that have diminished RNA binding clearly are functional otherwise. The mutations didn't mess up the protein. Now this slide here comes from 72 hours after transfection, which is this time point here. So here we are now measuring the import of the protein into the mitochondria. You can see the black wild type in the 3 subunits all have made it nicely into the mitochondria. But again, when we look at the earlier time points, and particularly at the 24-hour time point, you can see that all 3 subunits are delayed in the mitochondrial import. So providing in cellular evidence that RNA binding is important to facilitate import. This is another one. I'm looking on my clock, so maybe I don't describe it in quite as much detail. In essence, what's done here is the flip experiment. We have the protein, the wild type protein, we overexpress RNA ligands, either ligands that interact with the protein or controls. Here you see a bunch of controls, and they all don't change the pattern that you see here. But when we overexpress 3 of the ligand RNAs we've determined, you can nicely see that they increase the import of the wild type protein, but not of, the mutant that we compared it with. So what I'm showing you here then is yet another example of riboregulation, where at least the 5A subunit, which I showed you, I showed you it interacts with RNA on the mitochondrial surface, that mitochondrial import in vitro is promoted by the RNA. I showed you that the binding deficient mutants can rescue mitochondrial respiration, but nonetheless, these binding deficient mutants display delayed mitochondrial import into cells, and ligand RNAs can promote the import of the wild type protein. And hence, I think what we are seeing here is yet another form of riboregulation where RNA can foster import across a membrane, in this case, through the mitochondrial import machinery towards its final destination. Okay, so, these are not conclusions. This is sort of food for thought. So this example may give us ideas, or the field ideas, about the many proteins that are on this long list that have to undergo transmembrane transport, or that are transmembrane proteins, as a possibility of what the RNA interaction may mean. This we already discussed after my first part. That I think it's very important. We are well aware of the structural plasticity of RNA, and much of this riboregulation may be, physiological aptamers that evolution has selected rather than being selected in a test tube. If you want to be extreme, you could say that in principle, any protein actually could be a meaningful RNA binding protein, because you can probably make against any protein an aptamer. So at least to me, this riboregulation has at least the capacity to be a very early form of biological regulation. Because if you think about, for example, post-translational modifications, in order to regulate a biological event by a post-translational modification, you have to evolve an enzyme that specifically modifies the protein, recognizes it, is able to modify it specifically. In many cases, you need a machine that takes that off, so you have an on-off switch. Here in riboregulation, all you need is the target to be regulated, an RNA that can be evolved. Okay, so now to the second part. This is really ongoing work. It is work that, I have really thought whether I want to present it or not, and I've decided I'll present it for its interesting potential, implications. So this, what you see here, again, has nothing to do with RNA biology as such. You see, something that is also in the textbooks as a receptor-mediated endocytosis, classical piece of cell biology. The transferrin receptor is the molecule that's expressed on nearly all cells, often overexpressed on cancer cells that need to take up iron. So it is a cell surface receptor, which is a dimer. This dimer binds these blue eggs with red eyes. This is transferrin molecules, proteins that carry iron, the red eyes in the serum. And so, two molecules of this bind to the transferrin receptor. This triggers clathrin-mediated endocytosis. Then the endosome is acidified via proton pump. So the pH drops. This drop in pH leads to the release of the iron, into the endosome. So now the transferrin is turned into apotransferrin, ironless transferrin, as you can also see here. And this complex is then recycled to the cell surface at physiological pH of 7.4. Apotransferrin, unlike dipheric transferrin, has a very low affinity for the transferrin receptor. So it dissociates off to pick up new iron. And, the whole story begins over again. So the transferrin receptor was also on our list of RNA binding proteins. And Silvia Colucci, who is a current postdoc in the lab, who is investigating this, had previously worked in iron metabolism, and she absolutely wanted to work on this project. Why does this protein interact with RNA? So she carried out the assay that I already shared with you earlier on. Here you see the input. Here you see the 2C eluates. Here actually we have one additional control. So we not only do a plus UV, and you will see soon minus UV, that comparison, but we also do an RNase treatment or not before loading it onto the silica column. So the expectation is that prior RNase treatment, where you remove the RNA, that should result in the retainment of the protein, should obviously remove that ability. On the bottom here, you see two negative controls that shouldn't be RNA binders, and they're not. Here now you see two positive controls to very well-known RNA binding proteins, and you see they bind nicely after UV light and without the RNase treatment, but with prior RNase treatment, the binding is lost. And then here you see the transferrin receptor, and you can see the transferrin receptor sort of follows the pattern of the positive controls, supporting its RNA binding activity. But then when you look at the minus UV control, the known RNA binding proteins, they don't show any any binding anymore, as you would expect. But with the transferrin receptor, the picture has hardly changed. And is that RNA is sensitive? Yes, it is. So what does that mean? We use UV cross-linking, as I said, to establish a necessary covalent bond between the RNA and the RNA binding protein in order to have it retained on the column. This guy here doesn't seem to bother whether it receives this UV cross-linking. Might that mean that there is a covalent bond between the RNA and the protein already in the cell? What examples do we know of existing covalently bound proteins to RNA? Very, very little. We know of some viruses, like picorna viruses, that have their RNA genomes capped at the 5' end with a protein in covalent linkage. So that's an example. And then some of you may have seen a publication that appeared 2 years ago in Nature about RNA-ulation. So in RNA-ulation, the authors reported that in bacteria, in E. coli, that are infected by a T4 phage, the T4 phage introduces into the infected bacteria an ADP ribosyl transferase that normally, as you may know, uses NAD as a substrate for ADP ribosylation. But it can, this particular one, can also take NAD-capped RNA that had been described before and attach the whole RNA to these 2 ribosomal proteins that they had identified. So this is the only precedent that I'm aware of. And so the question was, could now this be a eukaryotic, a human example of where the transferrin receptor is covalently bound to RNA, something we have started to refer to as an RNA-tagged protein as opposed to an RNA-binding protein, or RTP. So this actually shows, again, the work that was published by Katharina Höfer and Andres Jeschke, who coincidentally come from Heidelberg. The 2 proteins that they found were the ribosomal protein S1 and the ribosomal protein L2 from E. coli. And as I said, an NAD-capped RNA substrate is covalently linked by the action of this phage ADP ribosyl transferase. And very nicely, that paper describes an NAD analog, a chemical analog, 3-methoxybenzamide, which is a competitive inhibitor of the function of this enzyme, and in fact is a competitive inhibitor of ADP ribosyl transferases that use NAD at large. And so Sylvia used that inhibitor, treated her cells with that inhibitor, and then looked at RNA binding of the transferrin receptor 1. And here you can see the blot, and here you can see the summary, the quantitation, and you can nicely see that this inhibitor, this is without inhibitor, and this is titrating in the inhibitor. That this inhibitor strongly reduces the signal by 2-fold or slightly more. Also, you may notice that even though from here to here, the dosage goes up by a factor of 20, there isn't any further reduction in signal. And at the moment, we can interpret that, but we don't know exactly what it means. But this data then supports the notion that also in mammalian cells, HH7 cells, the TFR1 protein may receive an RNA tag by this mechanism through an ADP ribosyl transferase. A second test one can do, which is also described in that previous paper on E. coli, is to attack the linkage after it has been established. So the previous slide was preventing the formation by inhibiting the enzyme. This is a chemical attack on that particular linkage using hydroxylamine. And so you can very nicely see here, this is the input proteins, and these are different concentrations of hydroxylamine applied to the lysate. And you can see here 2 RNA binding proteins, one classical, the PKM2 I referred to early on that we study, and there is no effect of hydroxylamine treatment. And you can very nicely see that hydroxylamine removes the ability of the transferrin receptor to be retained on these 2C columns. So all of this is in support of the transferrin receptor 1 being an RNA tagged or covalently, a protein that covalently binds RNA. Now, why did we pick it from the list? Because Sylvia was interested in it. She had previously studied iron metabolism, and so she picked this. Now, that's a pretty random choice, as it were, raising the question of whether there are other proteins in eukaryotic cells that might have a sort of similar function. And so in order to explore that systematically, we just applied a proteomic approach to this. So we again took HGH7 cells, which we UV cross-linked or omitted the UV cross-linking. This, of course, was the part we were most interested in. And then we would, prior to loading it onto the column, do RNase treatment or not. And so what we were interested in was this pattern here, where we had good RNA binding, even without UV cross-linking, which was sensitive to prior RNase treatment, so this would go down. Where when we do UV cross-linking, the signal doesn't jump up into the sky here, but sort of remains at a similar level as without cross-linking, and of course, it being sensitive to RNase treatment. So we ran the experiment, and the data that we obtained were sort of sorted into these 10 clusters. And you can see that cluster 5 and cluster 7, pretty much match the pattern that we were looking for. Maybe also look here at clusters 1, 2, and 3. And you can see that, here, the binding that's seen is mostly following UV cross-linking. So these are the categories, as you will see in a moment, that sort of have the typical RNA binding proteins. And we were obviously most interested in what is in these 2 clusters. Turns out that there's just over 300 proteins that sort of follow this pattern that are either in cluster 7 or in cluster 5. It includes the transferrin receptor, as we had expected. It also includes, amongst others, a protein called SCAM3. And I highlight it because we also had good immunoreagents against it, so we could use it for validation. And you see that here. So in this plot, you see a negative control, no RNA binding, typical RNA binding protein, HUR, the transferrin receptor you already know, and then here is a SCAM3. You can see when we do UV cross-linking that we get no signal here, signal with the normal RNA binding protein, and signal with both of these. When the pre-treatment is with DNA, so not with RNAs, and UV cross-linking, importantly, is omitted, then HUR disappears because there's no UV cross-linking. And you can see that this is still retained because it's not DNA sensitive. When we take RNase 1 instead, you can see that for both transferrin receptor and SCAM3, the signal disappears. If we take nuclease P1, which is both a DNase and an RNase, just to use a different form of RNase, you can again see it disappears, validating SCAM3. So, what do these clusters then look like? And here we just annotated the proteins that we found in the different clusters in terms of whether or not, or to what extent they contained previously identified RNA binding proteins. You can see that in cluster 1 to 3 in particular, almost all of these were previously identified RNA binding proteins. And you can again see that in clusters 5 and 7, only a small percentage was, well, only about half was previously known, but a large proportion wasn't previously known. What are they? Doing Go term analysis, you can see here the first 3 clusters. These are all terms, you can probably not read them, that are RNA processing, translation, et cetera, et cetera. When you look here for cluster 5 and cluster 7, looking at these here, you almost always see the term transport. And as you know, the transferrin receptor is, is a transport molecule. Where do they localize, these proteins? So these are the localizations of our classical RBPs as we know them. The nucleus, only 6% of these are in the nucleus. The cytoplasm, 5% are in the cytoplasm. The mitochondria, 3%. The ribosome, less than 1. So 85% are missing here. Where are they? They are along the endosomal pathway. So 31% being endoplasmic reticulum or Golgi apparatus, over 40% are the plasma membrane, 11% are in lysosome-associated proteins, some are in endosomal vesicles, exosomes. It's this pathway that is heavily enriched for these type of proteins. So that raises the question, first of all, again, how could this have been missed for so long? But perhaps more importantly, what is this telling us? Before going into that question, let me also say we next did the same type of approach, not using cultured cells, but using mouse organs or tissues, the kidney, the liver, the lung, bone marrow, spleen, and thymus. You can see a blue square when they turn out to be RTPs. Without looking sort of at the exact proteins, you can maybe notice 2 things. First of all, there is one particular organ that has the most blue. That organ is the liver. Incidentally, HH7 cells are hepatocytic cells, so there may be something about that organ and about hepatocytes. But you can also notice that there are, incidentally RTPs in other tissues found. There's no situation of where 5 or 6 are in one row. In other words, where a given protein is found as an RTP throughout, suggesting that these proteins are facultatively connected, covalently bound to RNA, and not apparently in an obligatory way. So also here, when you look at these from the mouse organs, it's exactly the same pattern as we observed before, relatively little in the classical locations and very much an enrichment of the endosomal pathway, suggesting that RTPs and covalent linkage may be important for this aspect of cell function and cell biology. So this is where we stand. I'd first like to state a caveat. The caveat for me is that while I think we have applied the controls that I explained to you, covalent RNA binding at this point has largely been assayed by association with the 2 C columns in an RNase-dependent and UV cross-linking independent way. We have to apply orthogonal ways of testing this in very rigorously challenging the notion that this bond is a covalent bond. However, I also showed to you, the usage of the ADP ribosyltransferase inhibitor and the hydroxylamine cleavage experiment, which very much are in line with that, covalent biology that was initially described in E. coli. So let me share, in finishing, some food for thought here. Obviously, one wonders whether RTPs may help define functional roles for mammalian RNAs that bear unusual metabolic caps. While NAD-capped RNAs have been described, there's not very much functionality yet sort of assigned to them. And in addition to NAD caps, there are other caps, metabolic caps. So that's a question to be resolved as we go on. Which are the enzymes that link the RNA tags to RTPs? The PARPs in mammalian cells are obviously prime candidates for doing at least part of that job. What do they recognize on the RNA and on the RTP for making this transfer? Do the tags remain throughout the lifetime of this RTP once it's attached, or is it removed afterwards? Sorry. Which enzymes would be involved in this, and in response to what signals? What do the tags functionally do? One of the aspects one can consider in terms of function is that we still have a very incomplete understanding of what explains protein sorting. I mean, lysosomal enzymes have mannose 6-phosphate modification, and it's a mannose 6-phosphate receptor that then helps transfer to lysosomes, but a lot is unknown. So does it play a role in protein sorting through these RNA tags? Is the RNA important for quality control aspects? What is the role in other organs and cell types? The central nervous system that has a lot of transport going on. We heard that this morning, how long the distances are that often have to be maneuvered in specific ways. Viruses, of course, take over their host cells by manipulating membranes of cells. So do viruses perhaps trick RNA tagging, and could that be exploited? Exosomes, many of you will know as vesicles that contain cellular RNA and are excreted from cells. The RNA that they contain are not sort of a stochastic collection of the transcriptome, but they're enriched for certain RNAs, and how exactly that is achieved is not known. And 50% of the proteins that we have on the RNA tag list are also exosome associated. And then finally, many disease proteins on the list of candidate RTPs. And so the question to be resolved is whether the RNA tagging affects phenotypes of some of these, and whether that could be used in the future. With this, I finish. I didn't have an acknowledgement slide after the first part, so let me just say, Andrila did a fantastic job on the ATP synthase. Sylvia is the key person with the story I just told you about, the RTPs, supported by Dunya and Pia. Pia also is the person who works on the PKM2 that I mentioned but did not show. Venki is doing a different project. Sudeep, Tom, and Tilly are our bioinformatics team. They often sort of get a bit of short change in talks given by experimentalists. And I find their work is just as important, and it's a fantastic team. Heike has supported us with the mitochondrial import assays. Bruno has also intellectually contributed very much to the RTP story. And with this, I finish and take questions if you wish.
Thank you very much, Matthias. We'll take a couple of questions. We have to get back today at 3:50.
This question is related to ATP synthase. I noticed that many RNAs related to ribosomes are interacting with ATPase, that protein, right? So do you see a connection between the major ATP consumer, which is ribosomes, and ATP synthase? Is there any regulation?
Yeah, so you make a reference to something that I did not mention, but that is in the bioarchives article that's been deposited. Of the 220 or so ligand RNAs, about half are top messenger RNAs that carry the top motif, that are largely messenger RNAs encoding ribosomal proteins. It's very interesting that there is a connection between the major source of ATP in cells and ribosomal proteins, or their messenger RNAs, which are obviously required in large number in proliferating cells. So whether there's a particular type of feedback regulation, that's what you're asking, is a very intriguing question. Unfortunately, I don't have an answer.
I have a question which is related. So ATP synthase will get folded in the cytoplasm, and during the transit the interactions might be taking place, but when it is going into the mitochondria, it needs to be unfolded. So is it that the RNAs keep it in a state which is unfolded, and then it goes in?
Right. So you're asking how mechanistically do we see RNA facilitating mitochondrial import? And you already mentioned one possibility, which is that it is sort of acting in a chaperone-like function, stabilizing an unfolded state of the protein that it requires to enter into the import machinery. That is a possibility. There are others. An alternative, not necessarily mutually exclusive one, is that around the mitochondria, some of the import substrates are, are increased in concentration compared to the rest of the cytosol. So maybe RNA could facilitate that concentration, local concentration of the import substrate near the import channel. That would be another possibility.
Thank you.
Could it be there is translation on surface of mitochondria, which can also explain why you have ribosomal protein also interacting with those RNAs? So you have ribosomal protein interacting with the ATP synthetase, right? So is it also getting translated on the mitochondria and then getting inside the...
Since the RNAs that are binding to ATP synthase are not the RNAs that are encoding ATP synthase, I would not necessarily see a translational mechanism, for the de novo synthesis of the ATP synthase. But again, a sort of a feedback control between ribosomal protein formation and import and formation of ATP synthase is perfectly conceivable.
That's the last question we'll take. Hi, over here. Yeah, so with regard to the RTPs that you mentioned, I just wanted to know your thought on what kind of RNA do we expect to be tagged to these proteins? Would there be specificity of the RNA in that aspect also?
Yeah, so I'm really sharing, as I'm sure you noticed, ongoing work with you. This project wasn't even started at the beginning of this year. So we are very eagerly now, on the one hand, trying to identify the RNAs that are connected, and on the other hand, trying to identify the amino acids through which the links are made, because that would allow us to do mutagenesis, avoid the linkage to be formed, and then follow the intracellular fate of those proteins, for example. So we don't know yet.
And thank you once again, Matthias.
Pleasure.